Three things motivate this talk. The first is biological risk from AI. So we heard a little bit about that from Soren and the International AI Safety Report. The second is the rise of AI agents. And the third is the evaluation gap. So the need for more and better measurement of AI capabilities and risks. That's where agentic biological evaluations come in. And as we, as researchers and a community, embark on the quest to build these types of evaluations, it's important to highlight how key design choices can shape meaning and the interpretation of evidence. Okay, this talk draws on a recent RAND perspective in which we lay out design choices throughout the eval lifecycle and map how they inform meaning. We hope this is useful not only to researchers who are designing evaluations, but also to funders and policymakers who are seeking to make sense of the evidence that evals provide. All right, so this is what we do. We organize the eval lifecycle into 5 stages: define, design, run, score, and document. And at each stage, we mapped non-exhaustively specific choices that shape what the eval is actually measuring. Importantly, these choices, which are often invisible in final reports, meaningfully affect capability estimates, risk assessments, and the downstream governance decisions that they inform. Okay, for the purposes of this talk, I'm going to use a case study by Kyle Brady and Jeffrey Lee and team at RAND. This is an agentic bioeval that just came out, and it measures whether a DNA or whether an AI agent can build DNA sequences. All right, let's get into it. So the first design choice I want to talk about is action space. Biological workflows span heterogeneous phases, from literature review to sequence retrieval, design optimization, troubleshooting, analysis. This is a bioweapon risk chain. So on the left, we have intention. On the right, we have release. And there's all these steps in between. And we have this iterative design, build, test, learn cycle in the middle. So an evaluation can target anything from the end-to-end capability to the localized capabilities in between, but specifying what the evaluation is measuring matters and where it fits into the broader workflow and threat model. The DNA acquisition eval that our team put together targets one step highlighted in red. So it's the design to build transition, asking again, can an LLM agent design DNA sequences for synthesis? Okay, the second design choice I want to talk about is human AI interaction and human actor assumptions in general. So most agentic evals implicitly assume a single agent just operating in isolation. That is typically not how agents operate in our world. They can be consultants, collaborators, or autonomous actors. But when a human is involved, there are also assumptions made about that human. So we make assumptions about the level and domain of expertise, about the resources available to the human, and making those assumptions explicit matters for eval interpretation and then operationalizing the results. So in the DNA acquisition email, the team outlines a specific persona. So this is a non-expert with undergraduate level biology, access to a benchtop synthesizer. And this profile matters because historically, a lack of expertise has been a key barrier preventing would-be threat actors from acquiring pathogens in that bioweapon risk chain. Okay, the third and last choice I want to talk about, it's actually two, but they're intertwined, is decomposing complex workflows and then developing robust scoring methodologies. End-to-end scoring or binary scoring can mask where failures arise and how they propagate. So decomposing the workflow into checkpoints can reveal where capabilities break down. This team in the DNA acquisition eval decomposed tasks and map scoring directly to those checkpoints, facilitating more granular analysis, which we'll see in the results. Okay, so where did this get us? The findings. So late 2025 models reliably designed DNA segments that met all the scoring criteria. So here we have models on the x-axis. We have the different steps. On the y-axis, we see like GPT-5, Claude Opus 4.5 got all the way through. Where the granular scoring comes in handy is we see the last two steps, the earlier models failed, but they got almost all the way through. So we can kind of see the nuances a little bit more in terms of what's happening beneath the hood. The team also physically validated O3's DNA sequence in a lab. Okay, so the takeaway from this is really interesting findings. We highlight that performance on a single task does not make a sweeping statement about the laboratory abilities of LLMs, but we do believe the results provide an indication that further exploration of this area is a worthwhile endeavor. And this really gets at how are we documenting evals and how we're communicating them. There's also points on info hazards, etc. Check out the perspective. I am just at time. One of these, so on all of the slides there, one of the QR codes is the perspective that talks about these design choices. The other is the research report. And also come find me after. We would very happily take feedback. Thank you.